Hello everyone, my name is Amit Chakraborty. I am a postdoctoral associate in BICE lab in Translational Dental Medicine, Boston University. Today I will be presenting my poster and also discuss the role of LSD1 in anti-tumor immunity and head and neck cancer. The title of my poster is LSD1 inhibition promotes anti-tumor immunity in head and neck cancer via interferon gamma, CXCL9 and CXCR3 axis and MHC class 1 upregulation. Head and neck squamous cell carcinoma or HNSCC have a poor prognosis, highlighting the need of new treatments. LSD1 or lysine specific demethylase 1, an epigenetic regulator, has shown promise in reducing tumor progression and increasing CD8 plus T cell infiltration. So aim of this particular study is to explore how LSD1 inhibition promotes HNSCC specific anti-tumor immunity and to explore how LSD1 can regulate MHC class 1 in tumor microenvironment to boost antigen presentation and cytokine response. To do that, we fed 4NQ to mice for 25 weeks and then we fed them SP2509 orally for 30 days as a treatment. And then we collected the tumor and we did qPCR, we did flow cytometry, then bulk RNA-seq and single cell RNA-seq to make the conclusion. To see the effect of LSD1 inhibition and how it promotes CD4, CD8 plus T cells and disease, we did a syngenic HNSCC mice model study. To do that, we have injected 4MOSC1 cells into mice tongue and developed the tumor. 4MOSC1 cells are also collected from the mice. They are actually epithelial cells collected from 4NQO treated mice tongue. So after injection of 4MOSC1 cells and after development of the tumor, we have treated SP2509 for 30 days. And then we did PCR and we performed flow cytometry to see the status of immune cells. With PCR, we can see that there is significant downregulation of KDM1A after treatment with SP2509, and we also can see that tumor size has been reduced significantly after 30 days of treatment. After 30 days, when we did flow cytometry, we can see a significant increase in T cell markers like CD4 positive and CD8 positive T cells. Also, there is a significant increase in conventional DC1, migratory disease and resident disease. So conventional DC1s, we have targeted XCR1 positive disease. For migratory disease, we have targeted CD103 positive disease, and for resident disease they are basically CD8 positive DC. After seeing the effect in syngenic mice model, we wanted to see the activation of dendritic cells in 4NQO induced mice OSCC model, where we have treated the mice with 4NQ water for 18 weeks and then after development of OSCC we have treated them with SP2509 for from 18 weeks to 22 weeks, and then we did our experiment to see the status of immune cells. In this particular experiment, we have given mice a low dose of sp2509 and high dose of sp2509. The low dose is 25 mg per kg and high dose was 40 mg per kg. So after the treatment, we have collected the tongue and did flow cytometry, qPCR, bulk RNA-seq and single cell RNA-seq. So in qPCR, we found that KDM1A is significantly downregulated after the treatment, as well as we can see that in high dose there is a very significant reduction of tumor lesions in compared to 4NQO treated mice. Then we did flow cytometry where we can see that CD4 positive and CD8 positive T cells have upregulated, and DCs are also upregulated in high dose of sp2509. Also we can see that CD86 positive DCs are upregulated. The CD86 positive stands for activation marker of the dendritic cells. After that, we performed bulk RNA-seq from 4NQO mice tongue tumor, and we found that after the treatment of sp2509, there is significant increase in cytokine signaling and T cell activation pathway related genes. As we can see that there is significant positive enrichment in immune response and T cell activation as well as cytokine activity genes. Then we wanted to examine the correlation between KDM1A expression and all the immune regulatory genes expression in TCGA data. We found that all the important T cell markers and cytokines are negatively correlated with KDM1A expression. Then with the help of single cell RNA-seq, we see that there is significant upregulation in cell to cell communication between T cell and epithelial cell and macrophage or dendritic cells after we treat the mice with sp2509. And in single cell data, we can see that there is significant upregulation in major genes that are responsible for MHC class 1 expression, like H2K1 and H2D1. We also observed that there is significant upregulation of TCR beta genes, and then we found that interferon gamma in T cell and CXL9 in DC cells also upregulated significantly. Then we wanted to verify how human PBMCs will react to sp2509 treatment in human cancer cells. To do that, we used humanized mice and injected them with human PBMC and HNSCC cancer stem cells. And we found that after treatment with sp2509, surprisingly, these tumors have reduced significantly, and all the T cell markers were significantly upregulated, like CD4 positive T cells and CD8 positive T cells. Then we can observe in KDM1A knockout mice tongue and sp2509 treated mice tongue, after KDM1A knockout or LSD1 inhibition, there is a significant upregulation of MHC class 1. We can now see that in blue. Then we also can see that there is significant infiltration of CD8 positive T cells in the KDM1A knockout mice as well as LSD1 inhibited mice tongue. We wanted to see the status of dimethylated H3K4 and H3K9 after LSD1 inhibition. We found out that after LSD1 inhibition, there is a significant upregulation of both H3K4 and H3K9 dimethylated histones. So after that, we wanted to see that these dimethylated histone status on MHC class 1 gene HLA-A. So we found out that there is significant fold enrichment of H3K4Me2 after LSD1 inhibition in HLA-A gene, whereas we can see significant downregulation in fold enrichment in H3K4 dimethylated version in HLA-A. So in conclusion, we can say that these results show that LSD1 inhibition enhances anti-tumor immunity in HNSCC by improving DC-mediated antigen presentation via the interferon gamma, CXCL9 and CXCR3 axis. Basically, MHC class 1 reacts with TCR beta and upregulates the production of interferon gamma, which then upregulates the CXCL9 production in dendritic cells, and that CXCL9 will bind with the CXCR3 of CD8 or CD4 T cells. Then those CD4 and CD8 T cells again produce interferon gamma, and that can cause the heightened anti-tumor immunity. This reaction, this pathway or this chain of reaction we can see in from our single cell data, and we can see it throughout all the models that we have studied. And also we can see that there is an increasing amount of MHC class 1 expression and changes in H3K4 and H3K9 dimethylated status on these MHC class 1 genes. So this mechanism may lead to new immunotherapeutic strategies for HNSCC. I am thankful to Dr. Manish Bais, my PI, for giving me the opportunity to work in this project, and I am also thankful to Dr. Maria, Dr. Stefano and Dr. Veralis for their support in this project. Thank you.